Peroxisomes. Peroxisomes are membrane-bound organelles in eukaryotic cells. They are involved in lipid metabolism and in the conversion of reactive oxygen species, otherwise known as ROS. Peroxisomes are also called as microbodies found in all eukaryotic cells. Peroxisome. Are they cell organelle? Peroxisomes are small membrane-enclosed organelles. They contain enzymes such as urate oxidase, D-amino oxidase, and hydroxylic acid oxidase, which are involved in a variety of metabolic reactions such as alpha oxidation of phytanic acid, conjugation of cholic acid. Can living forms live without peroxisome? Peroxisomes are responsible for oxidation reactions that break down fatty acid and amino acid. Peroxisomes chemically neutralize poisons through a process that produces large amounts of toxic hydrogen peroxide, which is then converted into water and oxygen. Lipid metabolism and chemical detoxification are important functions of peroxisome. How can we compare peroxisomes with living forms? Water plants, which function like peroxisome as both detoxify substances, that is water, alcohol, for their later use. Interesting facts of peroxisome. Peroxisomes are far simpler than other cell organelles like nucleus, chloroplast, mitochondria. The core of peroxisome is dense and crystalline. Peroxisomes are present in all eukaryotic cells, including human beings. Each human cell has around 500 peroxisomes except the red blood cells. Peroxisomes lack DNA and ribosomes. Do you know how the peroxisomes have originated and evolved? The origin of peroxisome has been suggested to be symbiogenetic, derived from an enslaved anaerobic hydrogen-producing prokaryote by D. Dewey in 1996 and Cavalier-Smith in 1997. Peroxisomes are synthesized in the endoplasmic reticulum, and the enzymes like urate oxidase, D-amino oxidase are synthesized by ribosomes bound to the granular endoplasmic reticulum. Who discovered peroxisomes? Peroxisomes, otherwise called as microbodies, were first described by J. Rodin in 1954 through electron microscopy in kidney of mouse. Peroxisomes were identified as organelle by the Belgian cytologist Christian de Dewey in 1967. Peroxisomes are devoid in prokaryotic cells. Peroxisomes in eukaryotic cell. Role of peroxisomes in eukaryotic cell. Peroxisomes exist in all eukaryotes ranging from unicellular protists to multicellular microorganisms such as fungi to plants and animals. Peroxisomes, which are membrane-bound organelle, occurs in the cytoplasm of all eukaryotic cells. Peroxisomes play a key role in the oxidation of fatty acids. Occurrence of peroxisomes in eukaryotic cell. Protist to name Saccharomyces cerevisiae. Fungi to name Coprinopsis cinera. Plant to name Capsicum annum. Animals to name Mytilus galloprovincialis. Location of peroxisome in eukaryotic cell. Peroxisomes are found floating freely in the cytoplasm in close association with endoplasmic reticulum, mitochondria, or chloroplast within the cell. Size and shape of peroxisome in eukaryotic cell. Peroxisomes are 0.2 to 1.5 micrometer in diameter. Peroxisomes are usually spherical in shape. Peroxisomes can change its shape and can be elongated or even form reticula in human cell except the red blood cells. Role of peroxisome in plant cell. Peroxisome play an important role in photorespiration in conjunction with mitochondria and chloroplast. Peroxisomes are the sole site of fatty acid beta oxidation in plant cells and are involved in generating two phytochromes, that is indole 3 acetic acid, otherwise known as IAA, as a signaling molecule, and jasmonic acid, otherwise known as JA, which is involved in the translocation of lipid intermediates. Role of peroxisome in animal cell. In animal cell, cholesterol and dolicol are synthesized in peroxisome as well as in the endoplasmic reticulum. Peroxisomes provide a compartment for oxidation reaction and is involved in lipid biosynthesis. Difference between peroxisomes in plant and animal cells. In plant cells, peroxisomes play a variety of roles, including converting fatty acid into sugar and assisting chloroplast in photorespiration. In animal cell, peroxisome protect the cell from its own production of toxic hydrogen peroxide. For example, white blood cell produce hydrogen peroxide to kill pathogens. Chemical nature of peroxisome. Peroxisome contain enzymes that oxidize certain molecules like hydrogen peroxide, which are normally found in the cell as fatty acid and amino acid. Catalase is found in the matrix of peroxisome and represents up to 40% of the total protein. The enzymes include urate oxidase, D-amino oxidase, and hydroxylic acid oxidase. Plasmalogens are the primary ether lipids in human beings. Structural features of peroxisome. Peroxisomes are one of the simplest organelles among the eukaryotic organelles. Peroxisomes are membrane-bound spherical bodies of 0.2 to 1.5 micrometer in diameter. They exist either in the form of a network of interconnected tubules called peroxisome reticulum or as an individual microperoxisome. They are variable in size and shape depending upon the cell shape and size and are usually found circular in cross-section. Peroxisome consists of a single limiting membrane of lipid and protein molecules enclosing the granular matrix. The matrix consists of fibrils or a crystalloid structure containing enzymes. The nucleoid containing peroxisomes are found in liver and kidney. Enzymatic role in peroxisome. The enzymes of peroxisomes are synthesized in ribosome attached to the rough endoplasmic reticulum. Hydrogen peroxide is highly toxic to human beings, animals and plants. So the enzyme catalase destroys it and probably play a protective role in all cells. The enzyme urate oxidase, D-amino oxidase and hydroxylic acid oxidase present in amphibian and avian peroxisome are related to the catabolism of purines. The enzyme catalase present in human liver helps in the breakdown of hydrogen peroxide into water. Function of peroxisomes. Hydrogen peroxide metabolism. Enzymes present in the peroxisome lead to the production and elimination of hydrogen peroxide, which is a reactive oxygen species, otherwise called as ROS. Photorespiration. Peroxisome in leaves, particularly in the green leaves, carry out the photorespiration process alone with chloroplast during daytime. Germination of seeds. Peroxisome in seeds are responsible for the conversion of stored fatty acid to carbohydrates, critical in providing energy and raw materials for the growth of germinating plants. Bioluminescence. Luciferase enzyme found in the peroxisome of fireflies help in the bioluminescence and thus aid the flies in finding its mate or its meal. The luciferase enzyme is also found in dinoflagellates. Bioluminescent dinoflagellate ecosystems are rare, mostly forming in warm water lagoons with narrow opening to the open sea. Lipid biosynthesis. Bile acid synthesis takes place from cholesterol of human liver. Peroxisomes contain enzyme to synthesize plasmalogens, a family of phospholipids which are important membrane component of tissues of the heart and brain. Disease caused due to defects of peroxisome in human beings. Impaired brain development and neuronal migration alone with myelin deficiency can occur. Skeletal system, liver, kidney, eyes, heart, and lungs are the organs affected due to defects of peroxisome in human beings. X-linked adrenoleukodystrophy, otherwise known as XALD, is a sex-linked disorder which can also occur due to the defects of peroxisome. Treatment of peroxisome defects. Cholic acid, otherwise known as Cholbam, is approved by the Food and Drug Administration (FDA), is administered for adjunctive treatment of peroxisomal disorders. Physical and psychological therapies are important for peroxisomal disorders.